Hello, my name is Michaela Fishback, and I will be presenting my poster for my research on population dynamics in endemic serpentine grassland plant communities amid anthropogenic environmental change. Serpentine grasslands are found on the hills around the San Francisco Bay Area of Northern California. These ecosystems are characterized by low amounts of nitrogen, high levels of heavy metals such as magnesium, as well as rocky soil and low water availability due to historically low levels of precipitation. These grasslands host 10% of the state's endemic species, though they cover only 1% of the landscape. Native species that have adapted to the uniquely harsh conditions in the soil are being threatened by the spread of invasive species due to nitrogen enrichment linked to high volumes of automotive traffic and air pollution in the area. When terrestrial nitrogen levels are higher, invasive, opportunistic species are able to come in and inhabit these grasslands and outcompete the smaller native species. Furthermore, annual precipitation is projected to increase in Northern California with progressive climate change that could further enhance the resource availability and suitability of these areas for invasive species. This research aimed to determine how the populations of native resource-conservative plant species respond to the availability of water and nitrogen in their environment when competition from non-native resource-acquisitive plant species is present. In order to do this, I recorded the seed production of a key serpentine grassland species under a range of water and nitrogen treatments at varying levels of invasive competition in a fully factorial greenhouse experiment. These two species are the native forb, Plantago erecta, seen at the left, and the non-native grass, Bromus hordeaceus, seen at the right. Plantago erecta is the primary host plant of the bay checkerspot butterfly, which is an endangered species that is found only in California's serpentine grasslands. An illustration is seen here. The seed production of a single Plantago individual was recorded at low, intermediate, and high nitrogen enrichment at both low and high water availability among low and high density Bromus. These 12 treatments seen here, along with the same 6 resource treatments for Plantago without competition, were repeated 4 times for a total of 72 plots. The figure in the middle shows the results of this experiment using a log ratio response for the number of seeds produced by Plantago. These results show how seed production varied from the baseline seed production by Plantago without competition, represented by the dashed line, with data above 0 indicating greater seed production and data below 0 indicating lower seed production than when Plantago had no competition. The left side indicates the results of the low competition treatments, and the right side indicates the results of the high competition treatments. Nitrogen treatments are shown along the x-axis with low nitrogen at the far left, intermediate nitrogen in the middle, and high nitrogen at the right. Low water treatments are shown in light blue, and high water treatments are shown in dark blue. So Plantago produced slightly more seeds under competition with Bromus than it did without competition when nitrogen and water availability were low, seen here. However, with increased resource availability, it produced significantly fewer seeds. This indicates that competition does not significantly impact Plantago populations if resource availability is low. These results also indicate that water availability reduces seed production of Plantago when nitrogen is low, but increases seed production when nitrogen levels are intermediate or high. This can be seen in the variation of the water treatments in the different colors of blue based on the nitrogen treatments at the bottom. Nitrogen has the greatest impact overall on Plantago seed production under competition pressure, as there is a clear downward trend in seed production at both low and high water availability at both low and high competition pressure. Ultimately, this experiment shows us that in order to protect these critical remnant habitats for California's native ecology, the impacts of increased nitrogen levels will be critical to mitigate as much as possible as the climate of this area undergoes projected changes. Furthermore, the conservation of these grasslands depends on understanding the combined effects of multiple variables in this ecosystem as a whole. That concludes my presentation. Thank you for listening. I would like to also thank my mentors, Elisa Hernandez and Dr. Lauren Hallett, as well as the rest of the Hallett Lab and the individuals at the Europe mini grant program for assisting with funding and supporting me in this research. Thank you.